All right, well, as I mentioned in class today, in this lecture we're going to cover the first half of Chapter 5, and then in class on Wednesday we'll actually cover the second half of Chapter 5. And so again, Chapter 5, what it does is it basically takes what was covered in the first 4 chapters, information about chromosomes and mutations and cell cycle, and applies it to Mendelian inheritance. Chapter 5 is about autosomal inheritance, and then Chapter 6 starts to explore some other areas of inheritance. Just a heads up, in a little bit, my wife's going to come back into the house. You'll hear the door open. You don't need to worry. Nobody's breaking into my house trying to hurt me. So let's get into the key terms. So this one has a lot of key terms. I'm sorry for that. There's really no way to avoid that for this chapter. So first term is a gene, and there are a number of ways in which you can define a gene. This textbook defines a gene in a way that I think is really helpful, and they basically just say anything that is transcribed into some kind of functional RNA is a gene. So this would take that number where sometimes you hear the number of 25,000 genes thrown out for the human genome, and those are just going to be genes that are transcribed into mRNA that ultimately is translated into protein. So this is going to extend the number of genes, and I think it's really helpful to think of a gene this way, because it illustrates all of the functions of the genome. All right, allele. The way they define this is just simply it's an alternative form of a gene. And so basically you're talking about an alternative form of something found on a very specific chromosome in a very specific location. Multiplex allelism. And so this is where you basically have a trait or a disease where you've got more than one mutant allele. And so it doesn't always have to be a genetic illness. It could be something like blood type, where you have 3 very healthy alleles present, or it could be a genetic illness like cystic fibrosis, where you could have hundreds of different mutant alleles alternating or altering that the protein. The cystic fibrosis transmembrane resonance receptor, or conductance, not resonance, conductance receptor. All right, complex heterozygotes. Complex heterozygotes are basically individuals that have 2 different mutant copies. And so typically these individuals are going to express whatever that genetic illness is, but they have 2 different mutant alleles. So functionally, they may be very similar to somebody who is homozygous recessive or has 2 copies of one of those mutant alleles, but they're heterozygous individuals with 2 different alleles. Genetic heterogeneity. Genetic heterogeneity is basically where you've got different diseases, a particular genetic illness that might be caused by multiple different genetic abnormalities. And so you can get what's called locus heterogeneity, where mutations at several different genes result in the same phenotype, or where you've got different mutant alleles for the same gene that result in the same phenotype. Codominance. Codominance we've talked about before in our crash course on Mendelian genetics, but I just want to mention this as far as what's going on at the molecular level. a little bit. So what's going on molecularly, at least with these genetic illnesses that have a codominance expression, is that both the dominant and the recessive allele will be expressed in these individuals. And so typically in examples of codominance, you'd have something that has a recessive inheritance pattern, or even a dominance inheritance pattern. And heterozygous individuals would have a slightly different phenotype than homozygous dominant individuals, in that they may have lessened severity of the genetic illness, or they may have just a different form of the illness than somebody who's homozygous dominant. And so there you're, again, in terms of what's happening at the molecular level, you oftentimes you have a normal functioning protein. If it's a dominant, if it's a genetic illness that's inherited dominantly, you would have the normal functioning protein, that's the recessive condition, and then you would have the abnormal protein product, that's the dominant condition, but both are being expressed. So you're still getting that normal phenotype, but you're also getting some degree of an abnormal function from having a new protein product. All right, monogenic inheritance. These are characters that are determined by a single gene. And in this chapter, it's really interesting because they define a gene as anything that is transcribed into a functional product, and then use the term monogenic inheritance and explain that it's not quite as simple as saying basically that a gene is something that is transcribed and then translated into a single protein product. And so monogenic inheritance are basically going to be genes, or sections of the genome, that lead or that are inherited under normal Mendelian inheritance patterns. So it may not actually be just a single gene. It could be multiple genes, but the alleles maybe don't assort independently, and so even though it's multiple genes, they act as a single gene. Or it could be multiple genes that do assort independently but still conform to normal Mendelian inheritance patterns and have a typical dominant condition and recessive condition. All right, the rest of these are going to be modifications to Mendelian patterns. Expressivity we've talked about, but in terms of disease, expressivity is basically going to, if you have something that doesn't express fully, you're going to get different levels of severity in the disease, even with the same genotypes. So this is the idea that where even if you have a homozygous dominant individual and it's a genetic illness with a dominant inheritance, you can still get different severities of the gene or of the disease in different individuals. Penetrance, again, this is one we've talked about before. Here in the context of genetic illnesses, it's basically if you don't have 100% penetrance, you can have individuals that have the genotype that should dictate a genetic illness, either homozygous dominant or heterozygous if it has a dominant inheritance pattern. And yet not all of those genotypes, not all individuals with that genotype actually have the genetic illness. And there could be a number of explanations for incomplete penetrance. A lot of them are going to be that there are maybe regulatory mechanisms that impact the expression of the gene. And so you may have a genotype that leads to a extra protein, but then it is told by methylation patterns not to transcribe, or it could be environmentally influenced. Complex heterozygotes. Complex heterozygotes, again, are individuals that that inherit two different mutated alleles of the same gene. Now, I want to mention it over here because it does oftentimes lead to alterations in Mendelian inheritance. And usually what you see that is that two parents that are carriers might actually give their offspring a a mutant allele at two different or at the same gene but two different mutant alleles, and then they end up producing an offspring with the genetic illness, even though that individual got two mutant alleles and it should be a recessive inheritance, but since they don't have a dominant allele with the normal protein function, they still have that genetic illness. Pleiotropy is where you have a single gene either controlling or influencing multiple phenotypes. This is fairly common with some of our genetic illnesses, where you have this protein expressed in multiple different tissues or multiple different organs. You can have a number of pathologies associated with the same mutation. Heterogeny. This you might think of this as the opposite of pleiotropy. Basically, heterogeneous, you've got multiple genes that basically mutations in any one of those genes produces the same phenotype. And then phenocopy. Phenocopy is basically the idea that because of environmental influences, you can have a gene that, or a completely different phenotype than what the genotype should produce. And so a good example of this is phenylketonuria, which is the inability to digest and process phenylalanine. And so with a diet that is very low in phenylalanine, you can avoid basically all of the pathologies associated with the genetic illness. Denovo mutations. Denovo mutations are mutations that appear in the life of the individual. They are not inherited, and they're oftentimes these are novel mutations. These are mutations that they might trigger a common genetic illness, but it's a mutation that oftentimes has never existed before. Lethal genes, these are genes that when you have a certain version of it lead to 100% or very close to 100% mortality in those individuals. And in lethal genes, if they are expressed before the individual reaches sexual maturity, these genes should be eliminated from the population unless there are a number of de novo mutations that trigger basically the same lethal condition. Modifier genes. Modifier genes are genes that influence the expression of another gene. Great example of this is epistasis, where you've got an epistatic gene that can mask the effects of another gene. This is an important explanatory factor for helping us to understand incomplete penetrance and low levels of expressivity. Are that many of these genetic illnesses not only have the gene that codes for the particular protein product, but then modifier genes that regulate that. All right, so monogenic diseases appear at the top of our chart. Again, are genes that are either a single gene or act as though it's a single gene. And in this table, I just want to point something out. As far as our genetic illnesses go, these monogenic diseases have some of the lowest prevalence numbers of any of our genetic illnesses. And I think there's an important point to make there. And the best explanation for why this is the case is that natural selection does a really good job of eliminating mutant alleles from populations. And because of that, these are not as common as genetic illnesses that tend to be entirely environmental, or not. If it's entirely environmental, it can't be a genetic illness, but that tend to be really heavily influenced by environmental factors, and genetic illnesses that are triggered by issues in dozens or even hundreds of different genes. All right. Well, don't know why all those came at once, but the first part of the chapter is really about dominant inheritance, or genetic illnesses with a dominant inheritance pattern. And so we have the need to kind of point out some of the differences between the mutations in those diseases that are going to have a dominant inheritance pattern and those that are going to have a recessive inheritance pattern. And so we'll start with recessive, since it just came up. Mutations that lead to genetic illnesses that are inherited through recessive patterns. These often lead to loss of function, where the mutation actually leads to the normal protein not being present. The mutations that most often cause these recessive conditions are nonsense mutations, where you get an early stop codon, and frame shift mutations, where you get an addition or a deletion that shifts the reading frame and just makes a completely different protein product, or no protein product at all. Where for recessive conditions, 50% of the gene activity needs to at least be sufficient for a normal phenotype. And so otherwise it would be more of a dominant inheritance pattern. So dominant inheritance patterns, usually these mutations are a gain of function. You make a protein that's never existed before, and it basically wreaks havoc with its presence. All right, these are often caused by missense mutations, where you change an amino acid into a different into a different amino acid and create a novel protein product. Oftentimes, these are mutations in regulatory genes, and so what you're going to see there is you create some novel protein product that is going to alter the way in which other genes are expressed. All right, so here are some common autosomal diseases. Many of these are a single gene, a monogenic inheritance. In fact, I think all of them are. And then it shows you the different genetic processes that are at play. And I want to show you just a couple of these columns to point them out. One, pleiotropy, where you've got a single gene resulting in a number of different phenotypes. And you can see that pleiotropy is really common in these monogenic genetic illnesses. And another one that's really common is multiplex allelism, in where you have multiple mutant alleles for the same gene. And so in both of these, it just really illustrates the need to understand the genotype of the individual in order to be able to make predictions on what you're going to see phenotypically, but then also to be able to understand what's going on genetically, and best counsel individuals with these genotypes on whether or not they should have children, and if they do, how to best prepare for the presence of a genetic illness. All right, so let's focus in on autosomal dominantly inherited diseases. Of the 6,000 or so monogenic diseases in humans, About 4,500 of them have an autosomal dominant inheritance pattern. The prevalence for these genetic illnesses is usually somewhere between 1 in 1,000 and 1 in 10,000 births. There are some that are more common, and there are a couple that are less common, but again, compared to other genetic illnesses, these tend to be very rare. Now, the severity of these genetic illnesses usually varies with genotype. And what I mean by that is homozygous dominant individuals usually have a much more devastating form of the genetic illness than heterozygous individuals. In fact, most individuals that are homozygous dominant do not survive long enough to make a contribution to the gene pool. Homozygosity is rare, and these can be caused or triggered by mutations in structural genes, Marfan syndrome and osteogenesis imperfecta. That should say imperfecta are good examples of this, and where you've got issues with connective tissues and issues with other and just organs all over the body as a result of mutations in structural genes. Also, you can see this in mutations in receptor genes, achondroplasia, familial hypercholesterolemia, polycystic kidney disease. In all of these, we've got a unique protein product showing up, changing the way that cells bind and respond to ligands. Mutations in some unknown protein gene. We talked about Huntington's disease previously when we talked about anticipation and errors in repeats. And one last point I want to make is this. Many of these diseases with an autosomal dominant inheritance pattern are only manifested in the presence of certain environmental factors. Many of these have very low penetrance, and the reason for that is that there are other genes that tend to mask the effects of these mutations, unless that there are certain epigenetic patterns in place, and those epigenetic patterns are impacted heavily by the diet of the individual or other environmental factors. And so this is going to be kind of a recurring theme for many of these genetic illnesses, is that it's not just nature of what the programming is, but it's significantly influenced by the nurture as well, environmental factors and epigenetic patterns. So as you prepare for class on Wednesday, keep in mind that we'll go real quickly through any questions that you might have on these autosomal dominant genetic illnesses, but we're going to spend the bulk of our time on Wednesday talking about autosomal recessive genetic illnesses.